Oil palm is the one of the plant that can survive more than 150 years and reach height of 30 to 40 meters. The annual oil palm is endemic to tropic and capable of growing healthy at optimum temperature between 29 into 30 degrees Celsius. Hence, grow oil palm under the assured irrigation condition by adopting recommended practice. However, the continuous use of agrochemical in this industry, such as chemical fertilizer, had led to several issues such as a serious soil degradation, nitrogen leaching, soil compaction, reduction in soil organic matter, and loss of soil carbon. By the uses of chemical fertilizer will also pollute the air and groundwater. Therefore, the use of microorganisms capable of promoting plant growth had been accepted as an alternative to reducing the use of chemical fertilizer in oil palm cultivation. In this study, the plant growth promoting rhizobacteria or PGPR had been transformed into a powder and will be measured toward to the plant growth. The soil sample will be using the spread plate technique using the glass rod, and then after 2 days, there will appear of single colony bacteria on the nutrient agar. After the isolation of the rhizopure bacteria, subculture and preserve the isolated bacteria with a different colony size, shape, and surface texture. Bacteria colonies in MH1, MH2, and MH3 have been appear in the nutrient agar plate. Bacteria in MH1, MH2, MH3 will be tested on the NPK plates. After the characterization have been done, the bacteria will be through a encapsulation technique, and then the beads will be incubated for 4 days to dry. Secondly, the beads had been grind using the grinding machine, and the last step is the PGPR will be get into a powder form. The growth performance of oil palm was observed based on the parameters such as plant height, number of leaves, chlorophyll, plant fresh weight, plant dry weight, root fresh weight, and root dry weight for a 5 week with a different treatment. Using one-way ANOVA, the significant difference in treatment effect was determined, and the mean of the treatment were compared using the Tukey-B test. Through the encapsulation technique, the use of sodium alginate powder is the option for the gradual delivery of microorganisms to the plant roots. Alginate is the polymer that has the greatest potential for encapsulation of organisms because the polymer chains can be linked by a calcium ions to form a bead. Using alginate to entrap the 50 bacteria and applying it into the plant is easily biodegradable in the soil and harmless to the plant.